Bacterial genetics. Before going with the genetics, so there are some definitions. Genetics, the study of heredity and variation of the reasons for similarity and differences between parents and their offspring. What is gene? The unit of heredity in the gene, a segment of DNA that carries in its nucleotide sequence information for a specific biochemical or physiological property. So it's a segment of DNA that carries the information. Genome means sum of total of these genetic information. DNA, you know, it is deoxyribonucleic acid, the principal molecule responsible for the storage of genetic information. Chromosome, the single largest DNA molecule in a cell that contains the information necessary for the cell's survival. Replicome, any segment of DNA that is capable of encoding its own replication in the cell. The chromosome is the single largest replicon in the cell. Plasmid, that is extra chromosomal DNA. It is capable of encoding its replication. In other words, non-chromosomal replicon. Chromosome, non-chromosome, or plasmid. What is phenotype? It is a characteristics expressed by a cell in a given environment that is adaptable in response to the environmental change. E is equal to GE. G is genetics, E environment, P is phenotype. Genotype, collection of genes encoding the characteristic. It remains unchanged, not like phenotype. DNA structure, I think you remember very much. So it's the phosphate, this is the backbone, and these are the bases, A, T, G, C, complementary matching. DNA replication. Bacteria have closed circular DNA. So genetic material in all organisms, E. coli, for example, 4 million base pairs. DNA replication occurs at the replication form. It from 5' to 3'. So just to total, this is the normal DNA. Now one CP, one form as leading strand, other is lagging strand. Usually leading strand form continuously, but lagging non-continuously. With RNA primer, DNA polymerase, it forms non-continuously complementary filling leading to formation of another chromosome. So this is the central dogma. You know, that is the pathway of protein synthesis initiated from DNA. It contains the message, or other way it's information. It goes via messenger RNA by means of transcription. Ultimately, it goes to produce proteins. This term translation. Similar thing happens in eukaryotes as well as prokaryotes. Just pictorial presentation, it's not much required. There are types of mRNA, you know, mRNA, rRNA, and tRNA. Translation, again 3 parts. It's ultimately leading to formation of a polypeptide or protein. So other terms about bacterial genetics, that is mutation. So definition, it is a heritable change in genotype. So I said genotype is fixed, but here heritable change in the genotype that can occur spontaneously or induced by physical or chemical treatment. Organism selected as reference strain is called wild type. So wild type is say no mutation. So polio, wild type used for other also E. coli we use. Their progenitor mutation is called mutagen. Wild type with a single mutation of the genetic material. Now it is a mutation. Process is mutagenesis. The agent-induced mutation is called mutation. So spontaneous mutation occur naturally. So part of natural selection, right? During DNA replication. So mutation rates 10 to the power minus 2 to 10 to the power minus 10 per bacterium per division. It can occur. So there are types of mutation. Just to, it's a recalling, like missense mutation, nonsense mutation, and silent mutation. Silent mutation, there is a substitution of one of the base pair, maybe several, but no apparent expression. But when we analyze the uh, gene material or sequence, once we doing the sequencing, we can see this. But others, they may express, leading to uh, maybe overproduction or underproduction or production of new proteins. Similarly, frameshift mutation, it's a large slide. Some there are lethal mutations. These all can be lethal anyway. And suppressor mutation and inversion mutation. There are, so it's a pictorial representation. Nonsense frameshift mutation, you can see nonsense and frameshift. All the frame is shifted due to one or two deletion or insertion. So this is the most important part. Genetic transfer in bacteria. Why it is important? One may, from their point of view, their survival, but for us as a clinicians, mainly pathogenic change or virulence modification. That's one. Other way, antimicrobial resistance. That is some key areas. So genetic transfer to the progeny can be classified as mainly it is vertical transmission, mother to offspring. But in here, in the bacteria, there are other way, that is horizontal gene transformation. That gone from one species to another species. But in the vertical transmission, it's the same species, mother to offspring. But horizontal transmission, it differs. It's from different species, inter-species transmission. So that is the important. So there are main three ways: transformation, conjugation, and transduction. Transformation is genes transferred from one bacterium to another as naked DNA, as a part, as a slice of DNA transfer. Conjugation, plasmid transferred one bacteria to another via pillars, sex pillar. Transduction, that is DNA transferred from one bacteria to another via virus or bacteriophage. So transformation, recipient cell must be competent to update the DNA. Recipient. Natural competence versus artificial competence is there. Only certain bacteria are naturally transformable, such as Streptococcus pneumoniae, Haemophilus influenzae, Neisseria gonorrhoeae, Vibrio. They are the most. So transformation takes place. DNA fragments from donor cell, donor, and the recipient. So recipient cell takes up donor DNA. Later it recombines or integrates into their chromosome and forms the offspring. That's how it goes. So this is a naked DNA fragments. So recipients, there are inherent characteristics. So it's not all bacteria are good recipients or recipients. So this is about transduction. Prior to that, about the you want to know about the bacteria parts. What I said earlier, page are viruses of bacteria. So you know Compared to virus, bacteria is giant. So bacteria goes in micrometer level, like viruses go nano, 10 to the power 3 minus 3 or 3. Variation is there. So virus reside in bacteria can give rise to lytic or destruction of the bacteria, or they may survive there. Its survival may be permanent. Then it is lysogenic. Otherwise, if it is temporary, so it is in between type. Like always lies. Temperature can stably infect and coexist within bacterial cell until the lytic phase is induced. So it's like intermediate. It's a type of viruses, bacteriophage viruses, lysogen. Page genome during lysogen is called propage. So that's page genome during lysogen. So page engulf DNA materials from one to other. So it now it's termed as propage. And bacterium cell is called lysogen. If the page genome encodes an observable function, the lysogen will be altered in its phenotype. That means propage carries genetic material, genetic information from one bacterium to other. It's a vector like kind of vector. So one example is diphtheria toxin. So once lysogenic transformation or conversion occur, as I said earlier, Corynebacterium diphtheria is non-virulent. But once this toxin transformation or production ability is gained, now it is capable in producing diphtheria toxin and become hypervirulent. That's how it occurs. So there are a few other terms. Can be transduction specialized or generalized, likewise. So specialized transduction. Normal page infect a bacterium with gene A, lysogen integration. Then some progeny page pick up them during the lysogenic phase. Now cell is, it contains A plus. Generalized transduction. It takes bulk like. Virulence and antimicrobial resistant genes can be moved by generalized transduction. That's the most important compared to specialized transduction. Specific transduction we can use as a mode of gene therapy. It will be a future good area. I mean, for the malignancies like leukemias, also metabolic diseases following kind of mutations, it's very much important. So that's what lysogeny and generalized transduction. So you can see this recombination happens, page, now it becomes propage, it transfer this material and recombination goes likewise. It's come here to go. Conjugation, that's the third one. Possible state of conjugation. Conjugating, it should be conjugating, permissive. Plasmid encodes all the function for conjugation and can move itself from donor cells to the recipient cell. So it mobilizable. Plasmid cannot move itself, but can be moved with help from conjugating plasmid. Conjugation functions. Synthesis of sex pillars, cell-to-cell contact via pillars. Copy in plasmid DNA and transfer copy into recipient cell. That's how it happens. So it's there. Donor, plasmid, via pillars, two pillars, there's contact, then plasmid is transferred to the recipient. So this electron microscopic picture. So conjugative plasmid are called donor, male. F plus. Bacterial receive the plasmid are called recipient, female. F minus. Likewise, it happens. So you can see it's a recombination here. Ultimately, they produce this recombined or plasmid DNA fragment into the chromosome. There are a few other ways: jumping genes, transposons, insertion sequence. So the most important thing is this is a mode of horizontal gene transfer from bacteria acqua, as I said earlier, resistant genes for the antibiotics as well as production of the virulence materials. Insertion sequence 1, gene encoding the transposition transpoase flanked by inverted repeated DNA sequence can interrupt genes if they are inserted into them. So that's how it goes. So thank you very much.